We have two speakers that have generously given up their Saturday mornings as well to talk with you a little bit about what they're doing in both the medical field and the agricultural field. And I can't say enough about how grateful I am that they are here with us. We have Dr. Kevin Flanigan from Nationwide Children's Hospital, and he is going to start us off with a few introductory comments and a little bit of background about what he does. Then we have Dr. Zack Bateson, who is from the National Agricultural Genotyping Center, who has been working with us for the last year or two, trying to help us make the connection stronger between activities on Grow Next Gen and the bioinformatics and biotechnology that's going on in his field. So he's going to talk a little bit, and then we'll open it up for questions. So without further delay, I'm going to send it over to Dr. Flanigan and let him give us a little introduction. Thank you again for being here, both of you.
Thank you very much for the invitation to speak. It's very exciting to be here. I've got a few slides I'm going to just call up here for a moment. I think this will work. I'm Kevin Flanigan. I'm the director of the Center for Gene Therapy at Nationwide Children's Hospital. I'm a neurologist by training, although my undergraduate degree was in music, and I love Dizzy Gillespie, so I really enjoyed the music that we were listening to, Dizzy Gillespie singing there. I'm going to do a few introductory slides, kind of quickly run through some of the most exciting stuff done in our place, and tie a little bit, I hope, to agriculture, because this interesting invitation to speak to you, there's not an enormous amount of direct overlap today between gene therapies and agriculture, but certainly genetic editing and genome editing will become more and more an important part in the agriculture field. But I'll start by talking about a disease called Duchenne muscular dystrophy. And very briefly, it's a disorder. It's an X-linked disorder, so it affects males and has onset around 3 to 5 years old. Boys by that time have difficulty walking. That brings them to our attention. They lose ambulation by about age 12 and die by the 3rd decade of life. And this is just a brief orientation because I'm going to show you some therapies we're developing. So this orientation will help. On the top here is just a cartoon of the gene, which consists of 79 exons. And those 79 exons encode a protein that's structure is important to remember briefly. It's got an amino terminus, a long rod domain of many repeats, and a carboxy terminus that binds to a protein called dystroglycan. What this does is at the muscle membrane, you can imagine that muscle fibers get distorted more than any other cells of our body compared to nerve cells or kidney cells or anything. Muscle fibers, muscle cells are under incredible stress with distortion of contraction. And dystrophin sort of is an anchoring, or many people call it sort of a shock absorber protein between the cytoskeleton and the membrane of the fiber. So when it's absent, that is really what we have is a leaky membrane and deterioration. Here you can see where dystrophin normally sits around fibers, and if it's absent, we get first degeneration and regeneration of muscle fibers. This is human skeletal muscle under the microscope. And with time, you get scarring, fibrosis, and fatty replacement of muscle. And that's what leads to the functional decline in boys. What we see is in an old textbook photo of 3 brothers, a young boy around the time of diagnosis, his brother who's just lost ambulation, and then his older brother after having lost, several years after loss of ambulation with scoliosis, and then respiratory and cardiac death. So we're very interested in fixing this and addressing it in our center. One of the important things to know as we talk about it is that there's a related disorder called Becker muscular dystrophy, in which mutations typically affecting the central rod domain still allow a reading frame that makes some of the binding to the cytoskeleton work, binding to the membrane work. And in that case, there's different shortened versions of the protein that are made. And you can see there's a lot of different versions. Each B here marks a Becker muscular dystrophy muscle sample. So various forms of these shortened ones can be very protective to muscle. And that's one of the key things to the gene therapy that we're doing now for Duchenne muscular dystrophy. How do we do gene transfer? Well, it'd be nice if we could just take a naked piece of DNA and put it into mammalian cells and have it work, but that's horribly inefficient. One nice thing, you can make a piece of DNA as big as you want, but it really doesn't work in muscle fibers. So instead, we use a virus called adeno-associated virus. And this has become the sort of stock in trade of our institution. It's a very safe viral family. These have no known disease association. And there's a wide tissue tropism. Many of these like to go to muscle or to liver or to brain. Different ones do it. So when these go into muscle, they don't integrate when they go into cells, they don't integrate into the chromosome, making them relatively safe. But one of the problems, and this is where we get back to the Becker muscular dystrophies, there's a limitation in the size of the transgene. So when we give these viruses, these bind to the cell, they go and are taken up in a vesicle, sort of with the protein coat then stripped off, and the gene itself is inserted in the nucleus where it stays and makes the missing protein. Now there are challenges to this, picking the right virus to go to the right place. Immunity to the capsid, we immunize people when we give them this therapy as a one-time therapy. I'll add one thing that's relevant right now to what's going on here in Ohio. Manufacturing capacity is a difficulty with this. Until the past few years, very few places have been able to manufacture these. It turns out that right now, Columbus, Ohio is something of a hotbed for this. Just in the past few months, a new company for manufacturing has been announced as a subsidiary of NCH. Another new startup company doing manufacturing has been started here in Columbus. De novo, and then the third company from Boston has cited their exploratory research and production laboratories here as well. So Columbus is becoming sort of one of the epicenters of manufacturing the AAV virus. So how do we correct Duchenne muscular dystrophy? How do we want to correct Duchenne muscular dystrophy? Here's that same cartoon, or a version of that same cartoon again. And it turns out that there are many ways you can take out large central rod domain regions of this and still have a functional protein. And in fact, 3 different versions of this gene are now in clinical trials. I'll show you just a very quick picture from a very recent publication from our center at NCH that shows in patients who are treated with this, we can get this miniaturized version of dystrophin expressing in muscle. Well, that's one of the things we're doing. One tie-in, I was thinking about how to tie this together with agricultural issues. And actually, much of another route to therapy are things that we learn, for example, from the agricultural or the veterinary world. So in addition to replacing a gene therapy, missing gene, we can try to modulate the function of that. We've done that. There's actually 2 interesting strains of cattle, the Piedmontese. There's actually several significant Piedmontese farms here, all of which have this incredibly muscled-looking phenotype. You can see this horse's rump, as they call it, on the Piedmontese cattle. Or, much rarer in the United States, the absolutely massive Belgian blue, hyper-muscled, double-muscled cow. I love this picture. I can't tell if this woman is particularly short or this bull is just really remarkably large, but it's an incredible amount of musculature, lean, very lean meat, difficult to raise in some respects, I think. But this animal actually has both of these breeds have mutations in a gene called, that it goes what's called ACTR2B, or ACTR2B. This is to inhibit muscle size. So in these strains of cattle, they have a mutation that makes this a non-functional receptor. There's observations from cattle and other animal models to say, could we make boys with muscular dystrophy better by making their muscles bigger? And in fact, one way to do this is by by delivering an inhibitor that inhibits binding of myostatin to this receptor called follistatin. And in fact, that trial has been done at Nationwide Children's as well, doing this in patients who already have some dystrophin, the Becker muscular dystrophy patients. So in fact, it does make somewhat of a benefit, although not as striking a benefit as we would hope to. I'm just going to close with a couple of comments on the very exciting new things of CRISPR-Cas9. I'm sure most of you are very aware of this. This system relies on a specific localization within the chromosome recognized by a guide RNA and the Cas9, an endonuclease that essentially snips both strands of when this happens, there's two ways that cells can repair that. One way is just to do non-homologous end joining, just with the DNA fragments joining back together, or homologous recombination, where a template of DNA is provided, and then there can be crossing over and repair. So these two pathways are active in mammalian cells, differently activated in different mammalian cells. But when we do CRISPR-Cas9 therapy, we're injuring DNA in order to let the cell kind of repair itself. And this is an enormously powerful tool, and I know Zack will mention some more about it in relation to plant agriculture. And I'll just point out, to use one more time that Duchenne dystrophy model here in dogs, this is a CRISPR-Cas9 approach to cleave out an exon and restore the reading frame. 49 fits with 52 here now in a way that allows expression. I'm afraid it doesn't project very well, but you can see expression here. Well, I'm screen sharing, aren't I? I meant to be sharing. I hope you can see here the dog in which expression occurs. Now, there's challenges to this, and it's important when we think about therapies. One problem is delivering this safely, particularly to muscle, about 70% of body mass. And one of our biggest worries with Cas9 therapies in humans is avoiding off-target effects. We don't want to nick a gene that has some other important role, like a tumor suppressor gene or something. So the other thing for us in the clinic, I do see patients in the clinic, is parents want to know, they read about it and say, when can I get that CRISPR right away? So I often steer them to a book, and since you guys are, your teachers, you're probably familiar with this book. If you're not, I would recommend it for any of your students interested in CRISPR. It's a co-authored by, one of the two authors is Jennifer Doudna, who's really, she won the Nobel Prize this year in chemistry for her work on CRISPR in mammalian cells. It is a terrific book written for the lay audience. In some ways reminded me of the double helix, which I know many of you would be familiar with, although it's fairer to her competitors than the double helix is. So it's really quite great, and I would recommend it to students. And one other thing I just want to leave you with about students is, for those students who are interested, at Nationwide Children's Hospital at the Research Institute, we have some wonderful programs for students, including the Futures Matter program, which is a paid summer research experience for high school students on our campus. Obviously we didn't do it this summer. I don't know whether we'll be doing it next summer, but for those students who are interested, it's really a wonderful opportunity to come work in a laboratory for 8 weeks, learn a lot of great science, and then present their work at the end. So with that, I hope I wasn't too long there, but with that, I'll turn it over to Zack.
Can everyone see my little laser pointer? Great. Well, good morning, everyone. Thanks, Dr. Flanigan, for starting us out. My name is Zack Bateson, so I'm a research scientist at a diagnostic lab. And so thanks for having me today, Jane, BioOhio, and Ohio Soybean. First off, I really want to thank you, teachers. It is because of you that I followed this career path in biology. I had awesome chemistry and biology teacher growing up. And I just appreciate all the hard work that you do to inspire the next generation of scientists, because we definitely need them. What I wanted to do is kind of share my briefly share my background and kind of my journey from wildlife interests into agricultural interests. And in doing so, kind of making the point that the biotechnology that I use in my graduate work, I apply to my postgraduate work in my current career. So for my graduate work, after I got a zoology degree, I dabbled thinking about going pre-med, pre-vet, but I just saw this great opportunity with research. And so I went into wildlife genetics. I was interested in studying small and isolated populations. And so I won't get into the details, but for my graduate work, I looked at 2 wildlife species, the 5-line skink, and then the endangered, state endangered in Wisconsin anyways, the greater prairie chicken. And the whole goal with these projects was to develop genetic tools to assess genetic diversity within these populations and apply it to some sort of management issue or finding ways to manage to boost the population. And so along these lines of using this biotechnology, I use DNA sequencing as well as bioinformatics to kind of explore that. Those same tools I was able to apply to my postgraduate work. The great thing about biotech is that when you're working on organisms, living organisms, they have DNA, RNA, those same biotech techniques can apply to anything that's living. So you can look at the pathogens, bacteria, viruses, fungal species. You can look at the host itself. So a lot of my postgraduate work, I work with honeybees and exploring the viruses, fungal pathogens that affect commercial hives in the US. I also look at diseases that impact our crops. And I also explore some of the weed issues that are occurring in agriculture, which I'll touch on in a little bit. So the company that I work for is the National Agricultural Genotyping Center. And so our goal is to translate scientific discoveries into solutions in production agriculture. And so we work with a lot of collaborators, both in the public and private sector, on various projects dealing with diagnostics. We are a small laboratory, so we're based out of Fargo, North Dakota, so I'm a little farther west than everyone there. We are an internationally recognized accredited lab, so we are ISO accredited. So we have certain quality assurance control mechanisms in our laboratory for being ISO accredited. And so what really motivates us is bridging this gap between the great research that's happening in academia and finding ways to apply it to the most fundamental level of agriculture, and that's the growers. And so there's a lot of great research happening at the academic level. An example of that is there was a lot of great diagnostic tests that I developed as a grad student. The problem is a lot of those tests kind of get lost after the grad project is done. And so what we do is we resurrect a lot of those tests and make them available to the grower. And so one of the big issues is that diagnostics is really lagging in agriculture, particularly plant sciences, compared to the veterinarian world or livestock world and human health. Most of the new research, the new diagnostic technology comes out of the human health field, and it slowly trickles down to the animal field. And then it seems like with plant sciences, it's kind of where the final step is. So we're kind of behind in terms of diagnostics compared to human health. And that's for, obvious reasons. But our goal is to kind of help boost these diagnostics in agriculture. And we really focus on pathogens and pests, mainly because they are one of the top threats to food supplies and food-based products. So in terms of what we do as a diagnostic lab, it really varies. And here are just some examples. So we do pathogen monitoring in soil. There's a lot of soil-borne pathogens that we can detect with the biotech that we use at the lab for soybeans, for other crops. Along with detecting the actual pathogen, we also use it to confirm disease in plants. So one of the issues is that there are multiple pathogens that will cause the same symptoms in plants. And the tough part is, in contrast to the medical field, the plants can't tell you what hurts. And so we have to really base our diagnostics off of symptoms, but then also try to find out what the causal agent is. The issue is different pathogens can cause a similar symptom on a leaf, for example, shown here. It could also be abiotic. So it could be just stress that could cause, a similar streak on a corn leaf or on a soybean leaf. And so we use our biotech to confirm suspicions on what the causal agent is. Another test that we have looks at ethanol contamination issues. So these include, there are bacterial species that cause contamination issues in ethanol production. So we develop a test to monitor those bacterial contaminants because if they get too high, they overwhelm the yeast that we want to produce the ethanol. So we have a way to monitoring that in ethanol industry. And then finally, one of our more current projects is looking at herbicide resistance in various weeds that impact agriculture. And so we have diagnostic tests that can confirm the species of the weed. There are weed species commonly called pigweeds that look very similar in the field. And so we have a diagnostic test that uses the genetics of these different weed species to identify them. But what more we're interested in is not only identifying the weed species, but also trying to figure out whether they're resistant to certain chemical control. And so this is a big issue in agriculture. So herbicide resistance, if unrestrained, if weeds are unrestrained in the field, there's estimates that crop yield would be reduced by 50%. And that's across US and Canada, so North America, it's about 50% drop. And so we're talking food as well. So a 50% drop in the food produced in the US, just based off of the interference from weeds. And the bill is huge. We're talking billions of dollars lost annually because of weeds in the absence of chemical control. And so I wanted to share kind of a story on the discovery of a genetic variant that causes herbicide resistance in these pigweeds. And so what started to happen in pigweeds, a species called water hemp, is that farmers were noticing that they were applying certain herbicides and it wasn't effective against certain weed populations of water hemp. And so scientists kind of explored the genetic basis of this herbicide resistance. And so what they did is they looked at the target gene for this particular herbicide. The group is called PPO inhibitor herbicides. These are used in soybean fields. And so they looked at the gene that they know that this herbicide targets. And so what I'm showing you here is an alignment right here. What I have is kind of the wild type, so the susceptible water hemp genotype on top, and then I have the mutant on the bottom. So I kind of have them sandwiched together here as an alignment. If you can see this small yellow arrow here, that's an exon in the gene. And so the circle is just showing you, I'm blowing it up here. So this is the piece of the exon. And if you look at the alignment, so again, the top is the wild type, the susceptible, and the bottom is the resistant genotype, you'll notice that there's all of a sudden a gap. There's a deletion that researchers found in the resistant water hemp population. And so if we kind of zoom in a little bit closer, it's actually a 3 nucleotide base pair deletion, which resulted in a loss of a single amino acid in this protein. So the mutant sequence has a deletion, the wild type does not. And so I'm sure a lot of you, ooh, fancy. I'm sure a lot of you, I forgot I put that in there, kind of wake you up. A lot of you teach this in biology, you know, structure equals function. And so when you have this loss, typically it's lethal. In this respect, the molecule is still active. So the molecule that the weed produces is still active. However, the target for that herbicide isn't quite exact. So these are just 3-dimensional depictions of that amino acid or the protein as it's folded. And so the circles, the red circles, are just illustrating where that deletion occurred in the protein. And so in the wild type, there's a pocket that allows for that herbicide molecule, that PPO inhibiting herbicide molecule to fit in there, rendering this protein inactive or having a reduced function. However, with that deletion, the protein is still functional, but that gap is larger where that molecule would fit, the herbicide molecule. And so the herbicide itself is less effective. And so again, we're seeing this deletion in the genome that causes this difference in the conformational shape of the protein, which then allows this weed species to be herbicide resistant. And so this is a big problem. I shared kind of a small story, but this is a big problem across the different herbicides that are used in agriculture. And so, you know, over herbicide use provides the selective pressure for weeds to evolve this resistance. And so in some states, the susceptible wild type is becoming rare. So it's becoming an issue in a larger distribution of the US and North America in general and worldwide. And so the problem is farmers have really limited herbicide options. There's really not a lot of new herbicides being released in the foreseeable future. And so the question is, well, what can be done to help? And so one of the more interesting pieces is using genetic engineering to insert into the genome herbicide susceptibility. And so Dr. Flanigan talked about the complications with actually doing this process, and it's such a new area of research, but there's already people thinking about it, trying to find ways of inserting susceptibility back into the population. And so this illustration is just showing you kind of how CRISPR technology, by inserting susceptibility back into this weed population, if we can get it back into the weed population, perhaps over time, we can start driving these populations from being resistant to herbicides back to being susceptible. So these chemical control options are still available to growers. So I think that's all I had for my talk. And hopefully you can kind of see how using this biotechnology, this CRISPR technology and other types of genetic engineering can be applied to both the medical field as well as the agricultural field.
Thank you both so much. I do want to kind of open it for questions, and I'm not exactly sure the best way to do that because we do have about 22 people other than us on the call. But if you want to put something in the chat, I think that would be probably the easiest way to do it. If people do have questions, I have a question to start us off. So as you are thinking about what you might want to ask either Dr. Flanigan or Dr. Bateson, if you would, I have a question for Dr. Flanigan, because when you said your undergraduate degree was in music, I need to know, how did you get where you are today?
Sort of backwards, came out backwards. So I was thinking, actually, I didn't acknowledge these great science teachers I had in high school back outside Joliet, Illinois, a little community outside Chicago. But my family was all musical. Everyone played an instrument. It's all I really wanted to do was play the trumpet. And we all went to the University of Illinois, where the music school is right next to the Morrow plots that Zack might know about, one of the older plots in the country, research plots for corn plot there. And about halfway through music school, I realized I loved science too much and loved sort of the interactions that I thought I would have as a physician. I went to the only medical school that accepted me and figured out during that time that I really got more and more interested in science. So had I really known what I wanted to do from the start, I probably would have done it differently and done an MD PhD program, or even given up medicine for PhD. But that's kind of, I fell into it backwards. I wanted to be a primary care physician, then I wanted to be a neurologist, then I wanted to be a neuromuscular specialist, and then I wanted to do genetics. So it was a long process, but just a kind of backwards one. But I still love Dizzy Gillespie all these years later.
Well, thank you for sharing that, because that seemed like a very circuitous path, not one that is traditional.
Well, can I just say one thing? I think it is, I have a lot of students who rotate through our labs. We have these summer programs and so forth. And I do encourage students to really make sure to explore broad things, because there isn't just one pathway now into either medicine or science. We have PhD trainees who really explored passions before they went into the PhD programs as well. So there's not just one route for the student who's really got a broad view of the world, I think.
Great. There's a couple people, well, actually one of our participants says that her son wants to pursue the trumpet and medicine as well.
Oh, is that right? Okay. Well, if he wants, feel free to email me, kevin.flanigan at Nationwide Children's Hospital, if he wants to talk about it. I'm happy to talk to him about it.
Great. And I do want to address that this presentation, we will be trying to make it available to classes. So hopefully you guys are okay with that we can allow these teachers to use those things in their classes. Another question for Zach, how to keep the susceptible gene in the population of weeds so it is not removed by the use of pesticides?
Yep, that's a great question. And one of the problems too is when you insert a gene, if it has some sort of fitness consequence, it's going to be quickly removed from the population. So people smarter than me are working on that, to be honest. So it's definitely going to be a challenge. And I think yeah, that's about all I got for an answer, I guess. Great question, though.
Well, I do think that, if we can think about this as teachers, to give our students opportunities to think through these kinds of issues is really what it's all about. I feel like sometimes we want them to think that their answer is this, and that is the answer, and then we're moving on. And what I loved about what both of you shared is that we have used chemistry and genetics to match things together, but there are multiple ways to do that. And so we're continuously looking for the best way, the most efficient way, the one that has the least deleterious effects. And I think our students are really under the impression that all of that's been done, that all of that has been decided, it's all solved, and it's just the way things are just terrible and they can't do anything about it. So to be able to give them some window into the fact that, and we're doing the same thing with the COVID vaccine right now. We're trying to figure out the most efficient way to deliver a vaccine that's gonna work. And that may be in one form or a different form, and we're gonna just have to keep doing the research to find it. And that's the way all of this is, I think. All of these problems get very small, when you're looking at those one genes or those areas of the genome, but they can make such a huge difference. Does anybody else have a question out there? Nobody has any questions. Okay, well,
I see one about muscles getting larger that I'm happy to.
Oh, yes, please do. Great, thanks.
So someone asked, are we seeing success with making muscles larger, and can this treatment treat other diseases like type 1 diabetes? So that's a great question. Actually, if you Google myostatin, you'll get 100 images of people related to weightlifters who are, you can go to GMC and buy compounds that are supposed to be myostatin inhibiting compounds and so on. So interestingly, I didn't show the results of that Becker muscular dystrophy trial. There were minimal changes in some of the fiber size measurements we have. In that trial, we didn't see enormous benefit, just some small amounts of benefit in walking with it. And one of the issues of doing systemic therapy for large muscles for something like diabetes there's other implications of how full a statin, where to increase the dose at a level that might be universally improving muscle bulk has impacts on other systems as well. But it's really a great question because there's an enormous amount of research going on in the community now about muscle metabolism related not only to diabetes, but to cancer cachexia. Patients with cancer lose muscle mass out of proportion to what's going on with their therapies or the cancer. There's other intrinsic factors associated with cancer that cause enormous amounts of muscle mass, which leads to disability and so on. So it's a major area of emphasis at Ohio State. Most of that has to do with other intrinsic pathways and genes that are more specific than myostatin's pathway itself for changing muscle function or muscle mass.
Other folks, ideas, questions, want to share anything?
Hold on, I'm trying to get this. I'm not.
Go ahead, Kelly.
I was going to ask with the new CRISPR 12A. I mean, with that range of precision, do you foresee that enabling more mammalian work?
So right, I really talked about Cas9. There's the 12A, Cas13. There's other approaches to using Cas proteins as well. And I gave sort of the really basic example of cleaving both parts of the DNA. There are other versions of Cas9, including Cas9 proteins that nick only one of the DNA strands, or ones that are called dead Cas9s, where they can be directed to specifically bind a double-stranded DNA, but not have the endonuclease activity. And those are being developed for targeting gene regulation as well. Yet another usage of it. So Cas12 and the other versions of proteins, I think we're just scratching the surface of how these are going to change. Many of the other ones, Cas13, for example, fit better in the AAV vector genome, which allows us the opportunity to think about making single products that might be more efficient than the way we designed these first round of things. One of the challenges, of course, is these essentially all come from bacterial systems. These are bacterial immune responses. So one of the challenges people are trying to sort out is in the mammalian context, how much will the Cas9s or the Cas proteins themselves generate immune responses, and what are pre-existing immune responses as well. So it's a great question. It's one we'll have a better picture of in a couple of years, but people are exploring multiple versions of Cas endonucleases now.
I had another question for Zack. I know your company works mostly with diagnostics, but in looking or learning about like Xanthomonas oryzae and its interaction with rice, and that whole theory that when we insert these resistant genes into rice populations in monoculture itself, it drives the bacteria to mutate so that it overcomes that resistant gene. Do you guys do anything on that with resistance, or are you all like diagnostics?
Yeah, we don't do any of that type of research. It typically requires you to have test plots and greenhouse space. We work with researchers that have that capabilities, and then we do kind of the diagnostic side of things. But yeah, I mean, you make great points, Kelly, about how monoculture in agriculture is a big concern. When you don't have that genetic diversity and you have an outbreak of something, everything's going to be susceptible to it. I mean, I'm talking in generalities, but that's a big issue. And the hard thing is that usually when it comes to virulence, it's not a one pathogen, one gene type of scenario. There's usually multiple genes that in the host that are related to the virulence of the pathogen. And so typically when you find these the linkage that I talked about, the herbicide stuff, that's a nice clean, here's a mutation that's associated with a phenotype. Usually it's multigenic, so it's very complex. And so when you're looking at susceptibility and resistance in the host for a pathogen, it's usually a large chunk of the genome that might be linked to it, and you don't really know exactly what gene is the major player, if there is a major player. And so to kind of sum it up, it's complicated, unfortunately, which everyone knows. Biology is kind of messy. So, yeah. So, I mean, you brought up a good example about Xanthomonas and rice, a bacteria, pathogen, and a host. And they're working on that, but that's always a concern, is that when you have a monoculture with limited genetic diversity and you're trying to find ways to cause resistance in the host plant, there's always this race, this evolutionary arm race between the pathogen and the host. And so you're always constantly at fight with that. And that's the same in human, disease too. We're constantly trying to find ways to overcome these pathogens. So
yeah, I know when they first started to figure out how Xanthomonas was interacting, and they found those tau effectors.
Mhm.
Tau effectors took a huge push, right? And they were, because they can modify or interrupt genes, right? They can disrupt. And they were starting to harness those for use, broader use, and then CRISPR came along, and that was old news.
Yeah. And well, and the tricky thing too with developing resistance in host plants is that typically you find something that you want to integrate into the host genome, or selectively breed, but that takes like an 8-year process, 8 to 10 years until you get whatever that resistance trait that you like in your experimental population to a variety that farmers can now use. The trouble is 8 years is a long time, and stuff changes, things move. And so the hope is with these genetic engineering mechanisms like CRISPR, that process will be a lot quicker. And so the plant pathologists don't have to wait that long until they can get something to market.
Can I ask one more question? I got one more. The corn that you showed with wanting to diagnose what disease was that you were looking at?
That's a great question. I don't know off the bat. It was probably Goss's wilt. Would be my guess. So a bacterial pathogen, but don't quote me on that.
Oh, but that's one of the ones that looks like another one, right?
Yeah, I mean, there's fungal pathogens that can cause streaking too. So
Yeah.
It looked a lot like Stewart's wilt, except for those little spots at the end, and I was like, "Oh, Stewart's wilt."
Yeah, and it could be Stewart's wilt too. So I mean, that's one of those troubles. Goss's wilt and Stewart's wilt is hard to differentiate. So, yep.
Other questions that folks have? We have about 5 more minutes. If you have something that you would like to shout out or ask or. Wait time. Okay. I have one other question. I thought it was very interesting that, Dr. Flanigan talked about the virus as a way to deliver, and that oftentimes in agriculture, we use the agrobacterium to deliver genetic modification. And then there's a Mosaic virus. Is that being used do you know if that's being used to deliver more genetic material than agrobacterium now? Because agrobacterium has some other types of, I don't know, side effects, maybe, consequences.
Not in mammalian systems. I don't know the Mosaic viruses well. Zach, maybe Zach.
Zach?
I'll tell you. I was going to say in mammalian systems, there's a big interest right now in engineering adeno-associated viruses. Adeno-associated viruses, although non-pathogenic, not associated with any human disease, they in high enough doses. We're giving doses in patients on the order of up to 1e to the 14th viral genomes per kilogram of patients. So these are extraordinary amounts of viruses we're delivering. Wow. So there is probably a limit where toxicity occurs that's not really specific. We don't believe we've hit that now, but the idea is if we can engineer viruses that have, let's say, a better tropism for the target tissue. So when we deliver something that's targeted to muscle, a lot of it is soaked up with every version of AAV is soaked up by the liver and doesn't have the function we want. So that's a big area of interest about engineering or doing what's called directed evolution to identify identify viruses that are more specific for the tissue we want. And then the other big thing in mammalian systems is non-viral gene delivery, which would include biopolymers and nanoparticle delivery. Was another future route, let's say, in the next years you'll hear about.
I would just like, if I could interject there about the delivery systems. I worked on a project where actually salmonella was used as a delivery system to solid tumors. And so my job was engineering out the pathogenic genes, and then they have something like a 10,000 times tropism to solid tumors, and then where they could be used to deliver the chemotherapeutic agents to the tumors. And it was pretty interesting, and the whole thing kind of fell through. This was at Yale. The whole thing kind of fell through when they did, in the mouse melanoma model, they discovered that the salmonella was pretty smart and left a rind around the tumor to protect itself. So just a little anecdote there. So
Yeah, I think it's pretty difficult to outsmart those evolutionary pathways.
Yeah. Oh, and just a comment too. I mean, this discussion about delivery mechanisms, I mean, to me, thinking back to high school, I would be amazed that we're able to take a virus and force it to do what we want to do, inject a gene, inject, various things into the cell. Same with like cloning, bacterial cloning, how we force those E. coli to take up a gene so we can study it more. I mean, to me, that's interesting to 16-year-old Zack, so it's really fun just kind of listening how that research has been evolving over the last, decade or so.
So we have two more just real quick questions, I think, hopefully. One is when you do pathogen ID, Zack, is that done with PCR?
Ding, 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 ding. Yep, PCR.
But then you have to have specific primers for those particular pathogens to amplify them, right?
Yeah, so the main technology we use is quantitative PCR. So we're able to take the soil sample or leaf tissue or whatever, amplify targeted pathogens, and not only say whether it's there, like a presence absence or a qualitative value, but we can quantitate it. So sometimes there's economic thresholds for some of these. So farmers know in their soil they have X amount of a particular soil pathogen, that there's something that now they need to do to manage it, whether it's growing a resistant crop or something like that. So yeah, PCR, and we quantify the causal agents.
Okay, and I think the last one is for you as well. You mentioned it was hard for the technology to trickle down to plant agriculture for obvious reasons. So as a non-ag person, Kathy Daniels is asking, what are those reasons?
Well, one reason that comes to mind is people are selfish. We want the technology for ourselves, right? Which is for obvious reasons. We want to extend our life expectancy. We want to solve problems with genetic disorders. And so that's where a lot of the research is at. I don't have the numbers offhand, to really support that claim, but all of the research and all of the biotech that I'm interested in started with human biology. So that's kind of what I meant. Sorry, I didn't explain that well enough, and I'm perfectly fine being the selfish species that all this biotech is started in people, or mouse models for people, so.
Well, I think, again, one of those things that's not so obvious is, okay, so they're weeds, so what? But when you can say to a student, these weeds are causing half of our food supply, if we do nothing about them, half of our food supply would be gone, that changes the scenario a little bit, I think. So, and I just need to throw this out there in terms of biology. We've gone from teaching plants as part of the kingdom of biology to not really talking about them at all, except on a cellular level. So I feel like that's a little bit of a misguided piece, that students graduate from high school sometimes not ever planting a seed in dirt or actually growing one. And that's makes me sad. So, but that's just my soapbox. Any last questions before we thank both Zack and Dr. Flanigan? Well, again, thank you so much